This is Seoul, Korea, and the Nansam Tower, and then this is the southern part. This is the northern part. There's a king, Garden of King. It's a garden party. This is Korea University. Is the entrance and main company. Today's topic is biological function of natural pigment on gastric ulcer and cancer. Nowadays is natural product is very important to treat some main goal is pigment and biofunctional material in nature. Nature have an important metabolic function in human. Especially pigment have some function antioxidant protective effect against disease such as ulcer and cancer. And in my lab is some isolate natural pigment. Astaxanthin is some rich in nature in shrimp or some green microalgae or red yeast. Especially in my lab is Xanthophyllomyces dendros is some red yeast produced some astaxanthin. So astaxanthin is a strong antioxidant against oxidative stress and some inhibitor tumorigenesis. And astaxanthin is structure is like that, and we isolate the mutant produced several overproduced astaxanthin, and some patient the mutant, and the culture condition as several compare the control ethanol or acetic acid or hydrogen peroxide is some overproduced astaxanthin. And usually nowadays is getting old. Old people is some suffer from arthritis. Some say is some induced ulceration. So we have a model system to using the naproxen and some 80 milligram per kilogram is optimum condition to produce the ulcer. Astaxanthin is I earlier mentioned about the astaxanthin antioxidant effect compared to the ascorbic acid is similar. So intracellular ROS scavenging activity is compared to astaxanthin. Ascorbic acid is astaxanthin is similar, and we are antioxidant effect, especially the protective effect of astaxanthin on naproxen-induced gastric ulcer, antral ulceration in rats. Here is a normal tissue, and we we occur the ulceration by using the naproxen, and we pre-treat astaxanthin, and then it's produce ulcer. This is a very good healing, and also is some lipid oxidation is some decrease, and SOD catalyst or glutathione oxidase is some enzyme activity is increased using pre-treat astaxanthin. And this is a therapeutic effect of astaxanthin on naproxen-induced gastric antral ulceration. This is some pre-treat the naproxen occur the ulceration, and then it's treat the astaxanthin. So that's why it's a therapeutic effect. is similar to healing the ulceration. Another natural product is some ulceration activity of anthocyanin. Anthocyanin from is rich in fruit or some vegetable, especially the rubus coreanus. Is similar the black rice berry. Regulate of the activity of matrix metal protein 2. We isolate from the black rice using the MS that the result show that cyanidin 3 glucoside and cyanidin 3 luteinoside is the main component of in black rice in our case. So it's similar to product effect of anthocyanin on nephroxin-induced gastric and for ulcer system in rat is similar to astaxanthin, as some is ulceration produced by the nephroxin. It's 80% or 70% of alcohol is a similar effector induced ulceration in rats. It's some healing effect. Similar to repeat peroxidation is some increase, decrease. SOD or catalase or glutathione peroxidase enzyme activities increase with treating the anthocyanin. And we checked the matrix metalloproteinase is important to healing to damage of the mucosa stomach, healing and damage. So we checked MMP9 to MMP2. So we treat the nephroxin as some activity of is a decrease, and we use anthocyanin treatment compared to nephroxin is some MMP2 is increase, dose dependent manner. We treat nephroxin is MMP2 activity is decreased, but we treat anthocyanin is increased. So another factor to TNF-alpha interleukin 1 better is some MMP2 is increased. Some the messenger RNA level is increased too. We used some anthocyanin treatment compared to the control. As some we treat 50 is compared to ascorbic acid is a similar effect. And next is some another anthocyanin source from the black rice brand. This is another the anthocyanin. NRF is a nuclear factor, E2 related factor 2. This is a natural pigment like astaxanthin or anthocyanin is some dissociate key band NRF2, and NRF2 is inside the nucleus, and then is some binding to antioxidant response element in a promoter region, and then is producing the antioxidant enzyme, so healing the ulceration. So we isolate the cyanidin 3 glucoside or ponidin 3 glucoside is the main component. Anthocyanin is similar to ROS or some lipid peroxidation or some healing like astaxanthin. Lipid peroxidation is decreased. Some catalase or SOD glutathione peroxidase activities increase. So NRF is increase treating with the pre-treating with the anthocyanin. So compared to another factor, pro-inflammatory cytokine is PGNK or 38 is a slight increase. NRF is some increase, significant. And we checked some NRF level, Western blot, or some immune staining is a similar result. And NRF is some binding of ARE2 and expression of NRF2 is increased. And then the main enzyme, the antioxidant enzyme, glutathione peroxidase activity is increased too. So this means it's antioxidant or astaxanthin or anthocyanin from black rice or some blackberry-like is dissociate keep from the NRF, and then as NRF is some binding to ARA, promote region of nucleus, and then is produce antioxidant enzyme battery induced, and then is enhanced protection against oxidative relative disease. And another factor, chiconin is some the red color root. In Korea is using this root and black color liquor is some wine-like liquor. So it's very edible, but this chiconin is very strong antioxidant, and then is anti-cancer. So we purify like NMR, and finally is chiconin, main component in a root. And usually cell damage is like DNA or oxidative stress. In this case, cell cycle is usually is G2, GS, G2M phase. G2M phase is some chiconin is some stop the AGS. We use the cancer, stomach cancer cell, AGS, and this mechanism is usually P21. Usually is a very antioxidant tumor suppressed gene is P53. Induced P21, and then is binding to specifics element is P21 promoter, activate the P33 independent mechanism in here. EGR1 is early growth response 1, zinc finger transcriptional factor, and then is EG1 is some induced P53, P21 or another factor. And so autophagy is now is a very important research field. Is we using the some cell cycle which part is some suppressed in cancer cell. So we G2 M phase is stopped this by the astaxanthin. Chiconin. So another pro-inflammatory and cytokine factor, cyclin D, or using the treat chiconin, cyclin D increase, or cyclin E, or some cyclin A is a decrease, H is similar. So we use the effect of chiconin on expression of cell cycle regulated protein in AGS, cancer, stomach cancer cell. P21 is the increase. Some CMYC, P27, P21 is some increase. Another one is similar. And we checked the EG1 with treat of chiconins. Some EG1 is increased with the treatment of chiconin. And then we checked the P21 and then EG1. So correlated P21 EGR1, we check. Here is a siRNA tweet, and then this is a control scramble. So this data set is some with treatment chiconin is increase the messenger level or Western blot. So chiconin is some usually is some induced EGR1. EGR1 is a binding of some P21 promoter region, and then induction of cell cycle arrest and cancer, stomach cancer, G2M phase, and then is inhibit proliferation against the cancer cell. Also, we used another. This is some yellow pigment, some natural product. Genopin is similar to research in chiconin. Is exactly similar factor. Some binding site or EGR1, P21, and it's a similar result. So it's not in detail. So it's similar to genopin is EGR1, and then is a binding site. P21 induction by the activated EGR1, and then is generation RRS, and then also autophase inhibit some stop the growing proliferation is a G2M phase, and then is inhibit proliferation against cancer. So conclusion is astaxanthin removal, repeat oxidase radical, and then potential remedy of gastric ulceration. Another anthocyanin or blackberry or some black rice is possible on anti-ulcer activity or induced activation of MMP2, attenuated activation of pro-inflammatory molecule. Chiconin promoter is promote and by transactivation EGR1 mediated P21 signal. Genopin is a similar one. This research is usually is Dr. Kim now is now moved to MD Aston to Berkeley University and postdoc position in now. Okay, thank you very much.